Maya and Ronen and all the students watching us, welcome back. In this segment, we will finally discuss the issue of building a tree to reconstruct the evolutionary relationship of sequences. Such trees are called evolutionary trees or phylogenetic trees. Classification of biological organisms and representing them in tree-like structure is an old tradition of biologists. The German biologist Haeckel is known for popularizing this representation, which integrated nicely into the theory of evolution as developed by Darwin at about the same time. This classical evolutionary representation used phenotypical properties of the organism, including jaw structure, bone length, number of teeth, etc., to build such trees. However, since we realize that biological organisms can be better characterized by their genotypic properties, including the sequence of their DNA or proteins, the field has gained great importance for a wide range of archaeological, anthropological, agricultural, epidemiological, and clinical applications. But let me start with an example from a totally different field. Look at these two texts in Hebrew. On the right is Psalm 133, as can be found in the canonical Bible. שיר המעלות לדוד, הנה מה טוב ומה נעים. On the left, you can see the same chapter as was found in the Dead Sea Scrolls, dated approximately to the beginning of the Common Era. It is amazing that modern Hebrew readers can still read this ancient text. When we read these texts side by side, we realize that they are not identical. There are spelling differences, but there are also different words and even different phrases in the two texts. The natural question to ask is which text better represents the original. The version seen in the Dead Sea Scrolls is much older, by about 1,000 years, than the oldest available handwritten text of the canonical Bible. Thus, it seems logical that the older text would be closer to the original. However, this is not the only option. As the scrolls may have been written by refugees who ran away to the caves in the Dead Sea from the Jewish center in Jerusalem, it is possible that they try to reconstruct the original text from their memory, a process that is likely to introduce many errors and deviate significantly from the original version. So can we use sequence comparison to determine which text is closer to the original? If we have only two versions of the text, we cannot. However, if we have many versions of the text, we can try to reconstruct their history, and this technique is as useful for biblical texts as for biological sequences. Let's look at these 7 biological sequences. We note that they are related but somewhat different from each other. Now, let's assume that one of them was the original, which then mutated along 2 branches, and then each one of the 2 resulting sequences accumulated mutation along 2 branches. Our challenge is thus how to place the 7 sequences on this tree. To identify the original sequence at the root of the tree, the 2 sequences that evolved from the root, and then the 2 sequences that evolved for each one of these 2. Actually, there are 210 different ways to map the sequences to the tree in different orders. And our challenge is to find the most probable scenario. Let's look at one of the possible trees. How many evolutionary changes must have occurred along this tree? If we look at only the left branch, we see that one change was needed to replace C to G, and then one additional change occurred on each branch. If we look at the entire tree, we see that 7 mutations were sufficient to create this tree. Note that if we assume that mutations occur at a relatively constant rate, we can also use the number of mutations along a branch as a measure of the time that passed since the split of the sequences at each node, supplying some kind of a molecular clock. If we try another one of these 210 possible tree arrangements, such as this one, we see that we need many more. In this case, 17 changes. In fact, in this example, the first tree that I showed you is the one with the smallest number of required changes, and thus we assume that it depicts the pathway through which these sequences evolved. Looking for the tree that requires the smallest number of changes, the most parsimonious tree, is one of the approaches to reconstruct the evolutionary process. There are additional approaches based on similar intuition that we will not present in detail here. Again, in cases where the number of sequences is large, the problem becomes computationally intractable, and thus all approaches can only provide a heuristic answer and cannot guarantee the optimal solution. We should note one more important point. In some clinical application, we may indeed have access to the entire set of sequences that forms a tree, from the root to internal branches to what we call the leaves, the external nodes. Such is the case when we follow the sequence of a virus from patient 0 to the entire population that he may have infected. However, in most evolutionary applications, we only have modern sequences without access to the ancient sequence of the root and the older branches of the tree. Thus, the challenge we often face is given only the current sequences to reconstruct the structure of the tree and maybe also the sequence of the ancient ancestors. In the following example, we have 4 sequences. What is the best way to construct a tree in which these 4 sequences form the leaves? In this case, there are 3 possible binary trees, and we can analyze all of them and find the ones that requires the minimal number of changes. During this process, we can also reconstruct the ancient sequences. You may have noticed that I only talked about binary trees in which each node can only split 2 ways and not into a larger number of branches. However, this is only a technical issue, as any branching pattern can be broken into a series of binary splits. An important question, however, is which DNA sequences should be chosen to build the tree. If we need to build a tree that will span different kingdoms of life, we should base the tree on a gene that is common to all organisms and is relatively conserved. So the sequence encoding ribosomal RNA is often used. However, when we want to build the phylogenetic tree of primates, for example, we cannot use ribosomes, since their sequence is almost identical in all primates, and there is not enough variation to allow for tree building. In this case, basing the analysis on genes evolving in neurological processes, for example, may be more informative. If we want to use phylogenetic trees to trace the evolution of human ethnic populations, we often follow these 2 points. First, to simplify the analysis, we try to base the tree on sequences for mitochondrial DNA, which is only maternally transmitted, or the Y chromosome, which is only paternally transmitted. So, what have we learned from building phylogenetic trees? To give just few examples, such studies have strengthened the view that life on Earth actually divides into 3 kingdoms: bacteria, archaea, and eukaryota. We know much more about the evolution of humans, including the fact that Neanderthals and Homo sapiens have interbred, and we can trace the history of different ethnic groups, for example, the peoples of the Southwest Pacific. Phylogenetic tree reconstruction is also used in many clinical and epidemiological studies, for example, in tracing the spread of the HIV virus in North America. Finally, we return to the question you asked me at the beginning of this unit regarding the physician who may have infected his own patient with the hepatitis C virus. Maya and Ronen, look at this figure from the original paper. As you can see, the tree clearly separates the patient who were involved in this case in red from other patients carrying the HCV virus in green. Note the very short branches between the sequence of the physician, patient number 1, and the sequences belong to the patients that he infected, suggesting that the doctor was indeed the source of the infection. Reconstructing phylogenetic trees is a complicated task, and we did not include many technical details, but I hope you understood the general principles behind the method and the wealth of evolutionary, epidemiological, and clinical information they can supply. This is the end of the series of segments on sequence comparison, but there is more to bioinformatics and sequence alignments. I look forward to meeting all of you again to discuss how bioinformatics can help physicians answer many more clinical applications.